All right, I'm going to go ahead and get started. I know you probably can't see me over the podium, but my name is Jamie Michalek, and I'm from FDA. I'm a senior product quality assessor in the Division of Liquid-Based Products 2. And welcome to session 4. Session 4 today will be about drug device combination and design differences related to quality and performance. We have 3 speakers lined up. Our first speaker will be Dr. Meenal Chavan. She is a senior pharmaceutical quality assessor in the Division of Immediate and Modified Release Products 3. That's within OLD-POPQ. Our second speaker today will be Captain Alan Stevens, who is the Assistant Director of Injection Devices in the Division of Health and Technology 3 within CDRH. And our third and final speaker today will be Dr. Rakesh Khilwani, who is the Principal Device Development Engineer for Teva Pharmaceuticals. With that being started, I'd like to welcome Dr. Chavan to the stage, and she will begin her presentation once it is loaded to the screen.
So good afternoon, everyone. My topic of presentation today is current regulatory perspective for demonstrating the quality and performance of proposed generic versions of transdermal systems, intravaginal systems, implants, and intrauterine systems. So throughout the day today, you have all learned about different types of the complex drug device combination product. I will be focusing on these 4 different dosage forms, that is transdermal and topical delivery systems, which are commonly called as TDS, vaginal system, implants, and intrauterine systems. Please note that there are approved generic versions of transdermals and vaginal ring. Till today, we have not received any official ANDA for the other 2 dosage forms, that is implants and intrauterine systems. And our thinking or our experience is only limited to the pre-development meetings and the generic development stage. So you have already learned a lot about complex drug device combination product. It can be any one of these. That is a co-packaged combination product. Here, the drug constituent part is contained within or co-packaged with a product-specific device constituent part. It can be either as a single entity or a co-packaged. There can be a cross-label combination product where the drug product is cross-labeled for use with a specific device in which the device design may impact the drug delivery to the site of absorption or action. Like the device design may meter the dose, and a single entity combination product, which is comprised of 2 or more regulated components, and the user interface may have specific use considerations. So in all 4 dosage forms, which I'm talking about, belong to the single entity combination product class. So in general terms, a drug device combination part is a combination of a drug constituent part and a device constituent part. For the purpose of this presentation, a device constituent part refers to the polymer-based components and materials which are used to deliver the drug constituent part of the combination product. The drug constituent part refers to the drug which may consist of just a drug substance or a drug substance which is mixed with specific inactive ingredients or a drug product. The review of combination product considers safety and effectiveness questions which are relating to the combination product as a whole, each constituent part and interactions between them. And therefore, some materials such as polymers may be considered during both the drug and the device review. So let me go in brief with all the dosage forms. So transdermal and topical systems. There are 2 main types of the transdermal delivery as a passive delivery and active delivery. The most common in the market currently are passive transdermal systems, which are broadly categorized as matrix transdermal systems and a reservoir transdermal systems. The examples of active transdermal delivery system include iontophoretic TDS or microneedles. So depending on the design, it can be targeted for local delivery, which are mostly referred as topical systems, or the systemic absorption, which are referred as transdermal systems. The vaginal systems, there are 2 main vaginal systems in the market as vaginal rings and vaginal inserts. Please note that vaginal inserts are not considered as a combination drug product, and thus for this presentation, we'll be only focusing on the vaginal rings. The vaginal rings are flexible polymer-based drug delivery systems which are for the release of one or more drugs directly into vaginal cervical tissue. Similar to the TDS, depending on the design, it can be targeted for a local or a systemic delivery. The implants are mostly categorized based on the route of administration, like nasal, ophthalmic, or subcutaneous. For the scope of this presentation, I am referring only to the subcutaneous implants as shown in this slide. These are again polymer-based drug delivery systems for release of one or more drugs placed subcutaneously and are categorized as parenteral dosage forms as per USP. These are long-acting dosage forms designed to provide continuous release of drugs either for a local delivery or systemic absorption. Intrauterine systems, commonly abbreviated as IUDs, is a flexible polymeric T-shaped device which is placed in the uterus. It's a long-acting dosage form designed to provide continuous release of drug for the systemic absorption. Let's briefly touch to Q1, Q2 sameness for the generic drug products. As per the USP general chapters, implants are categorized as injectable dosage forms, and hence Q1, Q2 sameness is required for the generic implants. However, for transdermal, vaginal systems, and intrauterine systems, Q1, Q2 sameness is not required by regulation. However, remember that the lack of sameness can have significant implications for demonstrating bioequivalence with the reference product. So to facilitate the generic drug product availability and to assist the generic pharmaceutical industry with identifying the most appropriate methodology for developing drugs and generating evidence which is needed to support the ANDA approval, the FDA publishes product-specific guidances or PSGs, which are describing the agency's current thinking and expectations on how to develop generic drug products, which will be therapeutically equivalent to the reference drugs. Specifically, in case of combination drug products, these PSGs are a good reference point for initial development with respect to quality recommendation. The specific and important quality recommendations such as Q1, Q2 sameness can be included in the PSGs as shown here. And we encourage the ANDA applicants to refer to the PSGs at a very early development stage. Let's talk about some ANDA submissions requirements. In addition to the regular information which is required in the ANDA submission, there are additional requirements which are required for the submission of generic complex drug device combination product. It can be separated in three main categories as a raw material, drug product development, and finished drug product. So the drug master file, which is known as a DMF, or device master file, which can be called as a MAF, for all constituents and LOS for the same should be submitted in the ANDA. During formulation development, the data from these studies such as residual drug, extractable, leachable, or human factor consideration shall be submitted. Additionally, comparative analysis with RLD using minimum 3 lots is highly recommended in the NDA submission. Special considerations for finished drug product are product design details, which shall include how the drug substance is incorporated with the device constituent part. For example, whether it's a matrix release design, whether it's a reservoir type system, or whether there is a drug which is solid core and contain in a ring. With engineering drawings as well as cross-section schematics are required. Instructions to patient about how to use the product. If the applicator or an inserter is used, then we need the engineering drawings of the same. Also remember that the stability shall be conducted on final fully packaged drug product. So these are the general requirements for all 4 types of the complex combination drug products, which are focus of my presentation. However, some of the critical attributes differ for each of these products, and this I will go over in my next few slides. Polymers is a very critical excipient for a combination drug product and shall be evaluated thoroughly. The polymers can impact the overall performance and quality of the finished drug device combination product. Detailed information about composition, characterization, residual solvents, residual monomers, as well as impurities shall be included in the ANDA submission. We recommend providing this information either in the ANDA submission or by reference to the respective DMFs. The manufacturing process for combination drug product is usually complex and can have a major impact on the quality and performance of the drug product. The manufacturing process shall be robust to ensure consistent quality and follow CGMP requirements as per the FDA guidance. For critical sequence like the skin thickness, we recommend continuous monitoring during the manufacturing. Establishing the impurity specification limits. Like all other dosage forms, the impurity limits in the combination drug products are assessed in line with MAP 5017.2 and ICH Q3B. However, as these drug products are specialty and unique dosage forms, special considerations are involved in setting up the specification limits of the impurities. In these dosage forms, usually the actual amount of the drug is high as compared to the theoretical amount of the drug delivered in vivo. As there is an uncertainty and limited data available about the migration rate and a penetration rate of impurities, agency's current thinking is to assume that the total drug load and impurities are released in a single day at one shot. And this is a worst case scenario. And hence the specification limits shall be based on the total drug content and not on the label delivery rate. In vitro release testing. For most of the combination drug products, FDA dissolution database recommends to develop an in-house drug release method, unlike other products. In case of long-acting combination drug products like implants or intrauterine systems, a real-time drug release testing is usually not a feasible option as a regular QC method, as the duration of action could be from months to years. Hence, we recommend to develop two different types of the drug release testing, as a real-time drug release testing and accelerated short-term drug release testing. So both these methods should be discriminating and robust. The accelerated drug release testing shall follow the same release mechanism as the real-time drug release testing. For transdermal and topical drug products, USP General Chapter 724 describes the drug release test using different apparatus as 5, 6, or 7. The end applicant can develop an in-house method, drug release method based on the USP recommendations. So now let's go with each dosage form in brief. For TDS products, adhesive is a critical excipient, and the adhesive performance will impact the overall performance of the final TDS product. The critical in vitro adhesive performance evaluation tests include peel adhesion, release liner peel, tack, shear, and a cold flow. We recommend other quality tests like heat effect, container closure pouch integrity, or durability and legibility of identifying label in the submission. The heat study shall be conducted on the RLD and a generic TDS to evaluate the comparative safety when it is exposed to the elevated temperatures. As per the TDS quality guidance, the identifying label is typically placed on the backing membrane of the TDS and should at minimum include the product name and the strength. It should be visible, it should be easily readable, and it shall remain visible throughout the duration of the action as well as after disposal. I'm referring here to critical in vivo studies for TDS, which are adhesion study and irritation sensitization study. It's recommended that the specification acceptance criteria or a specification limits for all these adhesive performance tests mentioned in the slide shall be for the commercial batches should be established based on the submission batches which are used in these two in vivo studies. For more comprehensive list of the test and data required, you can always refer to the TDS product quality guidance, which was published in November. The vaginal system. For vaginal system, the quality assessment considerations falls into three categories as mechanical characterization, other quality tests, and compatibility toxicology studies. Please note that this is a very comprehensive list and there may be more tests as well as more data required in the NDA submission. Complete biocompatibility testing as per the FDA guidance published in September 2020 is recommended for all generic vaginal systems. I've included the link here in my presentation. The implants, as mentioned before, as per the USP General Chapter, implants are considered as injectable products and hence product-specific quality tests as per USP Chapter 1 are required for assessment of implants. The other categories will be same: mechanical characterization and other quality test. Similar to in vaginal systems, for the intrauterine systems, again, the quality assessment consideration falls into mechanical characterization, other quality tests, and compatibility toxicology studies. So in summary, I will just say that for any of these complex combination drug products, you can always seek agency feedback via control correspondence or product development meetings. I would like to thank all my colleagues in OLDP and CDRH for their support during this presentation. And as mentioned earlier, we'll address all the questions during the Q&A at the end of the session. Thank you.
Hello, everybody. Good afternoon. I was a little worried about coming this late in the day, and then they sugared us up with snacks and Coke, so we should be good. But everybody after me is downhill. So do I have to push a button to get this to go? Oh, there we go. Okay. So I'm from CDRH, and I'm here to talk about the way that we review injectable devices that are part of the combination products. And we'll just go through it. So what I'm going to focus in on is primarily syringes and injectors, and talk a little bit about the performance criteria that we use to look at these devices. Mostly going to only be focusing on the engineering performance data that CDRH looks at. And so there's other, CMC or human factors information that's relevant for the device design as well, but I'm not going to be talking about that specifically. So I'm going to focus on these three categories of devices, and then I'll just roll through each of these. So for co-packaged syringes and needles, this is a type of combination product that we see generally. In fact, I'm pretty sure every example that I've seen, the device has already been cleared through CDRH 510K pathway. And these are the standards and guidance documents that we utilize for review of those products. But most of the time when we're looking at a generic combination product that has a co-package, the devices that are in that co-package have already been reviewed by CDRH. And so our assessment when we get involved in that product review is one, confirming, has it already been cleared? And therefore that satisfies a lot of the review criteria that we have for the performance data. And then secondarily, if that's true, is there any further processing of the product that's being done? So are you repackaging it from its cleared packaging that CDRH looked at, re-sterilizing it, et cetera? And that's pretty much all I'm going to say about the co-packaged products. So the rest of my presentation is going to focus on the prefilled syringes and injectors. So here's standards that are FDA recognized that we utilize for prefilled syringes. There are another part of the 11040, which is part 8 for the final product. It's not FDA recognized, but we do see a lot of manufacturers utilizing that part as well. And then we have a guidance document that we reference. And then for injectors, this set of standards is pretty robust in terms of the endpoints and test conditions and test methods for injectors. And this includes pen injectors, auto injectors, and with the 2022 version that was just published this past year, there's a new part for the on-body injectors as well. And just a quick note, the transition period for the previous version runs out in July. So moving forward, the 2022 version is going to be the recognized version as well. Okay, so when we look at the device, there's a couple of things we're looking at from a performance standpoint. One is the comparability of the device to the reference product in terms of its performance specifications. And then we're looking at the actual performance data as well. When we talk about in CDRH, the performance characteristics that we're primarily concerned about is for comparability is looking at those that impact the drug delivery function. So for example, from an auto-injector, the extended needle length, the injection time, the activation force, cap removal force could also be a key consideration depending on the product. And so these are the attributes that we look to see how does your product compare in terms of its specification range to the reference product. And then from there, we're looking at, do you consistently and reliably meet those specifications, for example, per the ISO standard. So it's two sets of questions. One, do you have the right specifications? And then do you consistently meet them? And that's what I'm going to talk about now in terms of the actual performance data that we're looking at. So one is, do you, given the specifications that you have, say for activation force, are you meeting that performance consistently? And so for injectors, the ISO 11608 standard is the primary method that we do design verification for these devices, as an example. And that standard provides a lot of robust test criteria, preconditioning, and endpoints. And these are key to consider. For example, and I mentioned the, assessing preconditions or in-use stresses, from an engineering standpoint, we're thinking about how is the product being used, what are the stresses that it's going to be subjected to in use, and we're expecting that the design verification studies are going to cover or simulate those exposures. So the ISO standard is one way of doing that for injectors. For prefilled syringes, I mentioned that the 11040-8 standard wasn't recognized. That standard does provide a lot of good information about the type of requirements that need to be met, but it doesn't go into the level of detail about for example, the in-use conditions that you might want to subject the product to. And then also, finally, the shipping and the aging studies, we're looking at having the device, having simulated shipping and simulated aging studies. And this topic is, I'll circle back to this at the end in terms of kind of lessons learned, best practices, but this is an area where we see frequent issues in the applications in terms of how those studies are conducted. Is it device only, or is it with the combined product? Ideally, we want the shipping and aging to be on the final product. There are situations where we see different data sets being leveraged, with the device only and then combined with the product. But ideally, those studies are going to be done with the final product. So I'm just going to walk through sort of the key performance criteria that we're looking at generally for syringes and injectors. So for functionality, we're looking for cap removal force, break loose force, glide force, and dose accuracy. For functionality, on the safety side, if there is a needle safety component, we're looking at the performance attributes of the needle safety device. That is a key issue. Biocompatibility and sterility are important. Generally, these would be covered as part of the CMC review. And so CDRH is generally not going to be specifically looking at those data for a prefilled syringe. Certainly for a co-packaged empty syringe, we would be assessing those criteria. And for needle safety, I skimmed past this, but earlier when I listed the standards, the ISO 23908 standard is the method that we use for assessing needle safety. For auto-injectors, I'm listing, the cap removal force, activation force, extended needle length, injection time, dose accuracy, and then any feedback mechanisms on dose completion are key considerations to look at. And then very similar needle safety, biocomp, and sterility. Again, the auto-injector is generally not part of the container closure, primary container closure system. And so The review of that is done in CEDAR. For the auto-injector body itself, CEDAR-H is looking at it for its biocompatibility of those materials. And so I would reference you to CEDAR-H guidance on biocompatibility. And we also recently released a guidance about biocompatibility for intact skin contacting materials, and you can reference that as well. So once we have the key performance for the functional performance for the device identified, we're looking at the design verification testing. The other thing we're interested for the functional performance is how are you controlling the quality of those specifications? And so in the application, you should provide a plan for the functional performance characteristics on how you will control the quality for the batches that you're releasing. And as with everything, we are expecting that it's a risk-based assessment. So what exactly is included in that control strategy is going to be directly dependent on how risky the overall product is. So it's not just the device, but it's the overall product. So from an experience standpoint, the type of things that we see that cause problems in the applications is, a missing comparison of your product to the reference device, as I mentioned. From a design verification and performance standpoint, is not having the appropriate statistical control on the performance data. So having that confidence and reliability data with your specifications is important. And then when you're doing shipping and stability, not including the functional performance of the device as part of those studies. So we routinely see, for example, stability studies and drug attributes are being assessed, but the device performance was not considered in those studies. Okay, and with that, thank you all for your attention.
Thanks, Captain Stevens. And this is a perfect segue to my presentation. I think we saw in the last slide that one of the best practices is to compare responses, device performance to the RLD. And that's what I'm going to focus on mainly in my topic. And that's one of the challenges that we face as the industry as to how to compare the RLD versus what we are proposing as a generic product. And how do we show that they are the same? They might have the same steps, exact same critical steps essentially. But when you look at the quantification of those performance, if they are different, how do we show sameness? So just a disclaimer, all the opinions and presentations are my own and does not reflect the opinion of Teva or its subsidiaries. And the scope here might be limited. So as an overview, background on the challenges when it comes to the essential performance requirements and how do we compare. I will use some case studies like a use case, a misuse case, and then there's a specific example of delivered dose because it is evaluated both from the drug side as well as from the device side. So it's a unique example. And possible proposals of what I think or what I think could be a good guidance that the agency can take and the industry can work together on. So when we look at the drug quality attributes, they're pretty well defined and applied in industry. However, based on the recent guidance on the combination products, the EPRs comparisons, there is pretty much a limited guidance when it comes to comparing the generic product to the RLD. And the first burning question that at least I always have is, what does it mean to be same? Is it okay if I have a small 10% difference, 20% difference? And as I'm making this comparison, and I'm coming up with a strategy of how to justify the sameness of the EPRs, does one comparison strategy works for all the EPRs? The answer is probably not, as we have seen for the whole theme of this presentation, it depends. And again, what is the best approach? Should we take a risk-based approach to comparing the EPRs? And can we separate them, like I said, as a use case versus a misuse case? And to see if we can separate them out and use different strategies for different performance requirements. So in general, when we think about developing a generic combination product, the first thing is we try to understand what the RLD has done. So look at what they have done, purchase some of the lots, characterize it, and essentially that sets the benchmark of what we are trying to achieve. But that's just a starting point. And then at that point, you also do not know what the specifications are for what the RLD has set during their own development process. So, and we also understand that unlike the drug, matching exactly the EPR performance one-on-one is pretty much unlikely to happen. Even a small design changes can be statistically different, may result in a statistically different output from the RLD. So it is very unlikely that there will be a one-on-one comparison between the RLD and the sponsor's product here. So that comes down to how do we set the EPR specification? And typically, if this is an innovative drug, it will be set on usability and human factors data. So it is good to also explore that. Can that be used also for the generic products, and how far we can use it? Again, it depends upon case-to-case basis, but it's at least a good starting point. And the last is it simply sufficient to compare the EPRs to the specification you have set, even though we see there is a noticeable difference between the RLD and the sponsor's product? So these are the questions that are still challenging, and I think there's further guidance required on that. So let's take a look at some examples. So I will mainly use a syringe with a safety device for this presentation. So in this case, let's use an injection force EPR. That is a use case. Every user is going to experience this force. If this is the kind of data I end up with, it's a slam dunk. No one is going to argue. My data is a subset of the innovator's data. Okay, that's fair, but the chances are unlikely. And again, these numbers are representative. It's not just for discussion purposes. So what if there's a difference of 20 to 40% in some overlap, but I'm well within my EPR spec? Let's attach some numbers to it. If the RLD has a mean force of 5 newtons, and if I have 6 newtons, is it meaningful? Probably not. Again, that would be an easy argument to make, but again, it'll depend on the product. But in both cases, I'm assuming that we are meeting our EPR spec that was derived from the usability and human factor study. And the main thing is that the user might not feel that difference. But here's a tricky one. I have an EPR spec, which I think is reasonable, but there is a 3 to 4 folds difference now. As a user, I would tend to assume that I would notice this difference. It goes back to, I think there was an example. If there's a 4-step auto-injector versus a 2-step auto-injector, if you're used to something, it might change your behavior. The question is, will it change my behavior if there is such a drastic difference that I still meet the EPR spec? The question becomes at that point is, if there is a change in behavior, is my EPR spec reasonable? That's the iterative logical framework that I would like to explore. But again, it's a challenging question. And the last one is even more drastic. What if there's a noticeable difference, but there's a discrepancy in the specification? Again, we do not know what the RLD spec was. And again, there's a difference in methodology. Whatever the innovator did, or the RLD manufacturer is doing, might use different test conditions. And they have set their specification based on those conditions. It might not match mine. So probably there is a room to say that we should not look at the RLD spec in a vacuum. But again, that a reviewer looking at it might not have the same opinion, and it might lead to a negative response or a deficiency request from the agency. So these are like some of the scenarios that we would encounter on when we're comparing or showing how the RLD behaves with respect to the generic product. And again, one example we can think about is, let's say there's an ocular injection, in which case one could argue easily that the 3 to 4 folds difference is unlikely to be agreed by the agency. But let's say if there's a subcutaneous injection, there's a chance there, because again, the risk is lower. So again, would be a risk-based approach, and it will depend upon the application. So this is where I thought it'd be good to propose something that one thing that we could focus more on is how we are setting our specification, because that's where all the risks as well as the usability data would feed into. And as long as that is being set appropriately, meeting that specification with good confidence and reliability would ensure that we have built quality into our combination product. To go along with it, we should also demonstrate in our submission that there is good process capability and adequate controls that are put in place to make sure that the submission batch and the production and the commercial batches would have the same quality. So there has been no process shift. And essentially, we are trying to mitigate the risk to the product quality using this entire process. So in the previous example, we saw a use case that every user will have to use the step and feel that will have to overcome that force to be able to apply the combination product. So let's think about a different case, that is the misuse case. So a safety system. A fraction of people will try to overcome the safety system. So let's say a user, for whatever reason, is trying to break that safety device. That will be a fraction of the users, and there will be a fraction of people who will be harmed by it. So the risk of that happening is low. And in that case, let's say if there is a small difference, it's easy to argue that because of the lower risk, it's okay to have these minor differences in performance. And it's also easy in such a case that as long as I'm above a particular threshold of the EPR spec, as long as the agency or the sponsor agrees that there is a good threshold of what we think that the user will not be able to overcome in the situation, as long as my performance is above that limit, it doesn't matter whether there is a 3 or 4-fold difference on this kind of a EPR, that is safety system override. What would still be challenging is if there is a discrepancy in the specifications between the RLD as well as the generic. That is another tricky area. And again, I think as long as the sponsor can justify that their spec has been derived from sound human factors and usability studies, I think there is, for this particular case, it'd be easy to justify that so long my performance is above that threshold, it is unlikely that a user will harm himself or herself due to this design. And the last case that I want to touch on, like I was saying, is the dose accuracy, that is delivered dose or volume. So let's take an example. There is an RLD that is labeled as nominally 1 ml. That's what the label claim is. So we go out there, we characterize the package and found that it is slightly less than 1. Let's put a number to it, 0.98. In a device space, or what we try to do is have a design verification activity as part of design controls. So setting let's say, no less than specification would be setting myself up for failure, because clearly the innovator itself has a less than 1 ml volume, and I would try to match it as closely as possible. So in that case, how does a sponsor set the specification? Should it match the RLD at that point, or should it still stick with the label claim? And is it okay to have different specifications for design verification with respect to release and stability? And based on my experience, I think this is one of the options that I think I've explored, is having a plus minus 5% acceptance criteria based on the label claim. This is in line with ISO 11608 standard. Essentially, you can still maintain your 1 ml target fill volume, but have a plus minus 5%. And for release and stability, still follow the USP standard of no less than 1.0 ml and use the rounding rules and essentially be able to meet the same requirement. So these are like some of the challenges that I have come across during my work at Teva. And I've seen some of these examples, and I thought it'd be good to present and initiate conversations on these topics. So I don't have a conclusion slide, so I think we'll take questions on the panel.
Thank you, everyone. I'd like to invite our panelists up for session 4. Then we can go through a quick introduction so you can put a name to a face. Right. All right. Meenal, if you want to begin by just doing a brief introduction, we'll go down the line.
So I'm Meenal Chavan. I work as a senior pharmaceutical quality assessor in the OLDP office, division of modified and released dosage products. And our team actually assesses all these complex dosage forms, which I was discussing, the transdermal, intravaginal, intrauterine, as well as implants. So that's.
Hi, Alan Stevens, the Assistant Director for Injection Devices at CDRH's Division of Drug Delivery, General Hospital, and Human Factors. And my background is engineering, and we review injection products.
Hi, my name is Garcia. I'm the Assistant Director of the Obstetrical and Reproductive Health Devices team at CDRH in OHT3. Our team is responsible for reviewing intravaginal systems, intrauterine systems, implants, as well as vaginal applicators.
Rakesh Khilwani from Deva Pharmaceuticals. I'm a principal device engineer and I have a mechanical engineering background as well.
Hi. It's not working.
Hi, my name is Yili. I'm a senior pharmaceutical assessor, and I'm from.
Yeah.
Green.
Right, both.
Oh. Yeah. Hello, my name is Yili. I'm a senior pharmaceutical quality assessor. I'm from OLDP, OPQ. So I'm in the branch. Basically, we review drug product quality of injectables most of the time. So, yeah.
Hi, I'm Ashley Boam. I'm the Director of the Office of Policy for Pharmaceutical Quality in OPQ in CEDAR. In addition to running the policy office where we help to work on a number of combination product issues, my background also includes a number of years in CDRH before moving to CEDAR. No, speaking to it.
Okay, thank you to our speakers and panelists for this session. With that being said, we would like to take some questions from the audience in person. Yes.
Yes. So there is one slide Stevens just showed about emergency used auto injectors, the reliability engineering requirements. I would like to know the latest thinking about that and like when this will be finalized. Thank you.
Yeah, thanks. So, I mean, we have the draft guidance that's published, and we're recommending that you would follow that if you have a product that falls within the scope of that guidance. And we're working through the comments now. I can't really comment about the timeline to finalize it, if that's what you're asking.
Yeah, my question is related to comparative EPRs. For some of the EPRs, there is a defined standard ISO guidelines and defined specifications like glide force, break loose force, and activation force. In that situation, does it make a sense that any differences in the test and innovator both are within the specification as per ISO? Then what would be the consideration? Because the question is like that, how much difference is called as a difference and is the same?
So from my understanding, the standards of a glide force that is for an empty syringe, I don't think about this an acceptance criteria for anything that is with a filled syringe. I think that is based on the user, how the user is going to use the device. So you set the speeds, you set the conditions. In that case, I think the iterative process, I think that's my opinion again, that setting the right EPS specification based on either what you find from the literature or from your human factors studies would be a good starting point. And then assessing the differences too, and whether those differences are meaningful.
Yeah, that's a good point. So I mentioned the ISO 11040 Part 8. That includes requirements for specifications like glide force, but it doesn't actually specify what they have to be or the methodologies. And so that's a challenge, right? So if you have information about the specification range of the reference product, you can use that to compare your product in terms of what the ranges are. Otherwise, you probably are doing testing to show that your performance and the reference product have similar performance.
So I really appreciated the question that Dr. Khilwani raised in his presentation. So I'm going to pose it to all of my FDA colleagues who are on the panel. If that situation that was posed arises where you have a reference listed drug that is supposed to be delivering exactly 1 milliliter of drug as the delivery dose, and in assessing it, a generic company determines that it's only delivering 0.98 milliliters, and they're trying to decide where to target. I was wondering if you could speak to, I guess, what the expectation is for dose delivery and what the acceptable error is for the reference listed drug, and then what the target, what that sort of average target should be for the generic company.
Yeah, that's a good question. So I think there's two parts of that. One is how are the performance specifications written? And so often for a product where you're, a single dose auto injector, the specifications are written as not less than, 1 ml or 0.8 ml or whatever the volume is. And so for that type of product, deviating below that would be considered a failure. So in the case that was presented, as stated, the ISO standard does have allowable deviations for the specification of, plus minus 5%. Again, that depends on how the specification is written. And assuming that the reference product has similar, performance specifications from an engineering standpoint, we would consider that to be acceptable. Whether that's acceptable to the CMC quality team in CEDAR is a separate issue as well that needs to be addressed.
Yeah, we don't see that case so often. So if the label says like deliver 1 mil, then the generic should be like have a specification say that deliver for volume is not less than 1 mil. But if you say, IOD like the deliver volume is less than 1 and inconsistent with the labeling, I would say, you should submit the information and we will review, say how. I haven't seen those type of cases like in reality. So really, if there's an issue like with the labeling and the real LD test result, we need to discuss further, and it's a review issue. And
we don't. I don't know that I've seen an example of that for a single dose auto injector. We do see for like variable dose pen injectors where you're dialing and you have different resolutions, you are going to get some variation around the set dose. And that's a lot of where 11608 is targeting the allowable deviation. So that is pretty routine to see that type of specification where you're getting a distribution around a set dose that the user is dialing. But typically for a single dose auto injector, it's written as a not less than specification.
Can I just ask a follow-up question on this? However, if let's say a sponsor does submit a data that demonstrates that it is less than 1, I'm assuming that will be taken into account during the review process and then the subsequent.
Yeah, I guess I would refer back to my other comment. I mean, that there's two sides. You may technically meet the standard, but if you're not comparable, if that specification is not comparable to the reference product, that would be a review problem.
Okay.
I have a question for Meenal. So during your presentation, you did mention that, the resolution has to be real time and accelerated methods to be developed, right? Many times we don't see in vitro, in vivo correlation in the real time resolutions, as well as when we develop an accelerated method, we see, the loss of discriminating power. Right, because we are doing force degradation there to accelerate the profile. So what are the bare minimum requirements and expectations from the agency's perspective?
I understand, like for these complex products, in vitro, in vivo correlation is not mandatory or required. And I agree, in the accelerated, sometimes you might see the loss of discriminating, but as far as possible, we always want a discriminating method, because that you are going to use as a regular QC method, right? So it has to be discriminating. So as long as you give that justification, but again, it's again a case by case. It can be a review issue. If you give provide a scientific justification why it's not giving a discrimination, the biopharm reviewer might consider that. So.
I'd like to take a break for just 1 second to introduce our colleague online, Dr. Ripen Misri. He's the Director of Co-Development Global R&D for Apotex. He's joining us online today as a panelist. So thank you. I'm sorry, Ripen.
Thanks for the introduction, and I apologize for not being there in person, but I'm pleased to have the opportunity to connect online. Thank you, everyone.
Hello. Yeah, my question is related to the comparative performance testing. For example, generic industry is developing an auto-injector and it's equivalent to the innovator. We have done the threshold analysis. FDA has agreed that it's equivalent to innovator. But in that case, whether we need to conduct comparative performance testing, because generic test device has to be comply with the ISO standard. Similar way, innovator has to be comply with ISO standard. Then what is the meaning of conducting comparative testing, test versus reference in that case?
Yeah, so that's a good question. So there are certain attributes that, if you have a reference product and your product that are compliant to the same standard, and the standard does have the quantitative endpoints or specifications you need to meet, that could be one way of demonstrating that you have the same performance. That's stipulating that all of the attributes that we're concerned about are covered by that standard. So for example, 11608 does include requirements for dose accuracy performance, but there are other attributes that we're concerned about that don't have quantitative specifications that are set by that standard.
For example,
like the extended needle length or the activation force, for example.
But activation force and, for example, dose accuracy, activation force, or break loose glide force, cap removal force, all those things are driven by ISO standard. Let us say dose accuracy. Dose accuracy is driven by ISO 11608 Part 1 standard. Innovator is meeting that standard, that's why it got approval. Now, test is also meeting and demonstrating that it is as per ISO 11608 Part 1. So is there any meaning to do the comparative testing for dose accuracy or break loose force, glide force, because it's driven by ISO standard?
Right. So I agreed that in that case, that's one way of demonstrating that you have same performance from an engineering standpoint. But there are some attributes that aren't covered by that standard, as I mentioned.
I would like to add to that. I think the ISO standard doesn't factor what indications the device is for, which may create a difference between what your pen is and what the RLD is. It will provide you guidance with reference to various attributes, but with reference to the risk, because of the indication, it does not provide that information in the ISO standard.
Okay, thank you. So do you mean to say that if there is no information with respect to tests which are primary functions and not the part of ISO standard, for that we need to conduct comparative testing? Is it like that?
Sorry, is that question for me? Yes. So just to elaborate a little bit more, for example, if you have a pen device which is being used by a general population versus a pen device that is being used for by a certain specific population, like patients with osteoporosis or patients who have some dexterity issues. So the type of, let's say, injection force or cap removal force requirements for that specific population would be different compared to a population which does not have any dexterity issues. So that is where you would need to demonstrate performance against the reference product, which is designed for that specific indication.
Okay, thanks.
Thank you.
Yeah, so I have a question. I guess I'll just open it for the entire panel. Excuse me. So Captain Stevens had mentioned very briefly that there's 11608 updated 2022 version that's available and goes effective July 9th for the transition. I was just wondering if the agency can elaborate on the expectations for pending applications and approved applications in terms of enforcement.
Well, I mean, I think, once the transition period has has passed, new applications, we would expect that you would be following the 2022 version.
Right. I think, the larger question is probably with applications currently pending, where that work has already been completed review, and whether or not we can expect questions to comply with the updated standard, and also approved products that may have had verification testing done, 1, 2, 5 years ago and haven't implemented any design changes. Expectations on complying with the 2022 standard and a lot of the standards that are changing in July for those specific application types.
Yeah, that's a good question. So I think what I would recommend is, specific to your question of post-approval changes, you will need to comply with the standard. I think the key differences obviously have to do with the identification of primary functions and the additional test matrix that's in that standard. In terms of whether or not we're going to be asking for that, if you submit like a post-approval change, it's going to depend on what the change is, because our review is going to be focused on that aspect of what you're submitting, in terms of whether or not we need to see that additional testing.
Okay, thank you.
Yeah.
So in use stability, we generally perform chemical testing and device performance testing. So like if we are performing in use stability for 28 days, so there is we perform testing initial, middle and end, like 0, 7, 14 and 28 days. So there is a chance that chemical stability may be changed. So that's why we perform 0, 7, 14, 28 days. But for essential performance required, can we do testing only on initial and end of the in-use date, like 28 days? Is it acceptable?
I'm sorry, you're asking for, can you repeat that last part of your question?
So in use stability, suppose my drug is used for 28 days, like pen injected, 28 days dose, right? So I have to perform in use stability, in which I have to cover initial, middle and end of the in use day. So for essential performance test, if I perform only 0 and 28 days, is it acceptable?
Okay, yeah, thank you. I understand your question now. I'm going to say maybe. I mean, I think it's going to depend on what the product is. we don't really have specific criteria on what the time intervals are. So that's possible. We generally do recommend that you have at least an intermediate time point if you're doing in-use stability. But again, that's going to, whether or not we want to see definitely require intermediate or more is going to depend on the product.
I agree with that. I think it's really important to have more than the beginning, the end, in particular, if you only have those 2 time points and we see a significant difference between the beginning and the end, that raises a question. And if you have that interim time point that can help provide additional information for why that's happening, that's a useful time point to have. So it's probably better to have more than just the beginning and the end in the assessment.
Yeah, I want to add a couple of things. So if your stability includes the functional test, and then the in-use stability study, you should test all the quality attributes in your stability specification. So in that sense, yeah, I think you should test all the proposed tests in the stability specification for the in-use stability studies. Unless, you provide the adequate justification, like why you eliminate the middle test, not like doing the full test of the stability specification. So.
Okay, thank you.
I have a question still on the reliability. So in the draft guidance, the applicant is encouraged to communicate the reliability method with the agency as soon as possible. So I would like, what do you think is the best practice to communicate the reliability method to demonstrate the reliability? Like what kind of elements should be included? And also what's the meeting type for this kind of communication? Thank you.
So yeah, I would recommend that you follow the pre-submission pathway specific to your product. And then we can communicate to you through that. So we frequently get requested to attend meetings with the CEDAR counterparts. And then the appropriate time is going to depend on your program, in terms of what your milestones are. So, the meeting goal, have established goal dates when we'll provide responses. I think, the type of information should be specific to your product and the recommendations that we make in the reliability guidance in terms of, where do you have questions about how to address that type of data and how can we help you? So the more information you have and the more targeted your questions can be, you're going to get better feedback from us.
Yeah, I would say a control correspondence might also be an option for you if you don't have other topics that make it worthwhile to pursue a pre-submission type meeting. A control correspondence would be another option.
So my question is with respect to the maximum daily dose to be considered for a longer, slowly released drug product, for example, transdermal patches. In such cases, such doses form, product is not being whole unit is not going into the systemic circulation. It is applied onto the skin. So the release rate is defined as a strength for such transdermal products. And even though RLDFY data says if for transdermal products wear period is for 6 or 7 or 8 days, you can consider a maximum daily dose by dividing the days to the content of drug divided by wear period. So what should we consider them as a maximum daily dose? Whether can we divide it by wear period or it should be a total drug content? Because PSG guidance also says that during a bioequivalence study, you have to do a residual drug content after bioequivalence study and compare with the RLD. And it proves that the whole drug is not going into the systemic circulation.
Yeah. So like I mentioned in my slide, right? So right now what we assume, that's our agency's current thinking, is that the total drug load and the impurities will be released at once in a single day, whether it's a wear period of 7 days or whether it's a wear period of 30 days. That's the reason when you're setting up the impurity specification limits, we are considering the worst case scenario. So you have to use a total drug load, whether it's for impurity specifications or extractable leachable studies. Because the main assumption behind that is we do not know the penetration rate of the impurities. There is very limited data available. And hence, that's what is the agency's thinking. So you have to consider total drug content.
Okay.
Do you want to take a question from online?
All right. So there are a few questions online. There are two questions regarding this topic, and it's for Ashley. When is the FDA draft guidance on EPRs expected to be released?
Wouldn't we all like to know the answer to that question? We don't have an estimate. All I can tell you is it's a clearance, and hope to see it published very soon. I know Ellen hopes even more than I do, I think. So
That's true.
Thank you. There are a few questions around this topic. Maybe Alan, Monica, and Yili, you can attempt these questions. Any guidance or protocol reference around transportation study? Do we have any guidances or protocols? And the second question is, in shipping study, is vibration and drop is acceptable, or we need to perform actual shipping study and then do the analysis to show the equivalency?
So there are a number of recognized simulated shipping and distribution and handling standards that could be referenced to do those assessments. I think depending on the type of product that you have, there could be some ability to do kind of not all of it, but just some of it. I think it just depends on the product and the risks of, or how fragile the product is and how important that shipping and distribution is. But there are some recognized standards out there for that. And Alan, anything else to add?
Yeah, the only other thing I would add is that I would pay attention. There are different ASTM standards for the simulated shipping, and I would pay attention to make sure you have the right one. And some of them are for like single unit packaging, and some of them are for pallets. And for some reason lately, I've been seeing a lot of products being tested according to the single unit shipping, when that's not actually how the commercial shipping is going to be conducted. So it's important that how you simulate the shipping is consistent with how you intend to commercialize and ship your commercial product.
Okay, there are a few questions regarding the validation. So first question is, do we need method verification or validation for functionality parameters? Is there a method validation guidance document for functional tests?
So specific to vaginal systems, intravaginal ones, no, there's not guidance. There are some for copper IUSs out there. There's, I think, one recognized standard there. So for us, I think a lot of the things that we look at when we look at performance testing for these products, particularly for the intravaginal products, we are in some ways kind of the Wild West. We just have to look at the protocols. We are able in some ways to rely on some recognized standards for diaphragms that we can leverage information from, particularly for intravaginal rings. But when we're looking at the validation protocols for companies when they're assessing the mechanical performance, we do look at the protocols, make sure that they're appropriate and that they've tested the appropriate number of samples, as well as we make sure that the acceptance criteria are something that's clinically relevant. We've talked about this a little bit earlier today. It is really important from our perspective when looking at these products that are inserted vaginally, that they are the forces that are used to input them or to remove them are clinically relevant and are not something that would be impossible for someone to do. So those are things that we think about when we think about that kind of testing.
Okay, there are a few questions for Alan. So the first question is how to carry out ASTM D4169 for DDCP, which needs to be stored about 2 to 8 degrees Celsius temperature.
I'm sorry, how to follow that standard?
How to carry out ASTM D4169.
Right. I mean, that's one of the shipping standards that we were referencing. I'm not exactly sure about the question. I'm assuming it has something to do with the storage. So, I mean,
yeah.
If you're deviating, I don't have the standard in front of me. So I'm assuming that the question's implying that they're deviating from the standard in some way.
With the storage temperature 2 to 8 degrees centigrade.
Yeah, right. So if you're the key, I think, from our standpoint is that you're simulating how your product is going to actually be shipped in the conditions. And so if you're deviating from something that's in a standard, explaining why and how and whether the way that you're doing it is having any effect on the endpoints from the standard, I think are key things that we would be looking for.
Okay, the next question is, in regards to dose accuracy with glass pre-filled syringes, is there a better approach than following ISO 7886 test methods, which are called out in ISO 11040?
So that is those two standards are the ones that we frequently see. We don't have a better method, if that's what's being asked. Granted, as I mentioned, the 11040 Part 8 doesn't have endpoints or methods specifically for those attributes. And obviously the 7886 standard is an empty syringe standard. So we do see manufacturers adapting those test methods specific for their products. But I don't have any other methods to point to.
Next one is, in light of 11608, 2022, the introduction of primary functions. What are the expectations for mechanical driven injectors, future use versus EPRs? Going forward, it seems primary functions shall be applicable only.
Yeah, so as mentioned, we have a guidance that's forthcoming on the EPR topic. There definitely is a lot of overlap in how 11608 defines primary functions and how our pending guidance is going to talk about EPRs. And so I think that manufacturers are going to have some choices in terms of what they're defining as primary functions and EPRs in a marketing application. Obviously, they can set them. It's a little difficult to talk about this because I don't have a guidance that everyone can look at and read and understand. And so I think there's going to have to be more conversation about this moving forward. But ultimately, what it comes down to is what is the data that's needed to be submitted in an application? And if there's differences between what 11608 defines as a primary function and what FDA defines as an EPR, what does that mean in terms of the type of data? Well, what it means is that you're going to meet 11608 for the primary functions, and that for the EPRs, you're going to be providing design verification testing for anything that would be considered an EPR above what's in ISO. But that's only if there's differences.
Okay, thank you. How the introduction of primary functions in ISO 11608 for mechanical NIS will change FDA expectations on essential performance requirements, considering the differences between both. Going forward, do we expect for such systems only primary functions are applicable?
Yeah, so I think I just answered this.
Yeah.
Again, it is a little awkward to have that conversation when we don't have a guidance.
Okay. If the method is not validated through ISO reference, we can go for in-house QMS established. Maybe Monica or Rylan.
Yeah, I think if you have an established method for, like I said, a lot of our products, we don't have a recognized standard for assessing a number of the functional or mechanical parameters. So I think an in-house method is acceptable for at least our products. I won't say that for Alan's products, but for the intravaginal products, it's reasonable.
Yes.
Thank you. Next question is for Rakesh. Is there any reference available for finalizing the limit for brake force and glide force for the PFS system without device, as this is one of the EPSRs considered by FDA, or this needs to be established based on multiple lot data in-house, as during development, we are not sure what speed RLD has generated that data?
So that is a good question. We never know what the RLD did. So the onus is on the manufacturer to understand what the use case is here and doing some clinical research as to what the right speed is. Looking at the simulated use case helps you get to that answer. And it's a really simple approach to get there. It's not something that cannot be done. As far as just to clear out the terminology, when I think about standards, a lot of standards talk about glide force. That throws me off because glide is mostly on empty containers. It's more extrusion force. And again, from the understanding of the question, it seems it's coming from a drug person rather than a device person. They're thinking in terms of drug manufacturing, where they narrow down the specification after making multiple lots. So that's again might take you into a different directions rather than think about usability and the use case. That will put you on a path to get you the right specification, and then you can start the development process after that.
Thank you.
Can I just add to that? Sorry. So it might be useful to think about some of this in the context of design controls, which do apply to these combination products. And I'm going to defer to my expert colleagues from CRH down here, but just to remind folks that there is an expectation that is as part of GMPs for combination products that Part 4 is going to apply. And part of that requirement then is the design control requirements from 820. And basically the idea is that, as you're developing your product, you were thinking about what they call design inputs in terms of, what the uses are going to be, who's going to be using it, what are their expectations. If it's a patient and something's be self-administered versus a trained healthcare provider, all of those, the use cases you've heard a lot, folks make reference to, you need to think about who's using it. You should be doing that as part of your development. That is an expectation as part of your design controls. So you should then always be able to point back to that. So even as you make changes, potentially going forward, you come back to those original design inputs and design outputs and to make sure you're still meeting what you originally set out to do. But let me ask my CRH colleagues if they want to add to that. But I just thought it might be helpful to remind folks this is an expectation under GMP as part of your development process.
Yeah, I mean, I 100% agree. And I think in the spirit of generics, a lot of what we're looking at from a design validation perspective is how does the performance of your product compare to the reference product? And so, to the question about injection speeds, in part is how does that compare to the reference product? And if there's a question about, what is the right speed? Some of the things that I think about is, when you're doing your method development, varying the speed, having different speeds that you're testing, and are you seeing a change? And is that the same type of change that you would see for the reference product?
Yeah, I agree. I think, those design controls are there for a reason, and they're really helpful when you're assessing the specifications of the device, or you're making sure that the inputs and outputs are correct. And I think when, like Alan was saying, when we think about comparability, especially for our products in the kind of intravaginal OB type space, we have a lot of flexibility in terms of what we consider to be comparable or not, because we're not expecting for these products to be completely identical, particularly with mechanical performance, because there are a lot of things that can change the mechanical performance of a product. How you cure the polymer can affect it, what kind of drug is in it, where that is manufactured, all those different things can in fact affect the mechanical performance. And so we have an ability to make a decision on what we think is a threshold that is not acceptable for performance versus what is. And it doesn't have to be, it's one of those things that's kind of nebulous. You can't explicitly state if you meet X, you'll make it. You have to look at the data in its totality to make that decision on if it's comparable or not, at least from the device point of view.
All right. Thank you, everyone.
Maybe time for one more question, and then we'll head to the concluding remarks.
Okay. So the last question, how EPR are derived for pen injection? Is it through understanding the IFU of RLD or from ISO 11608? Because ISO points about only technical requirements of design. So the question is how EPR are derived for pen injection?
Yeah, that will all be explained in the guidance that we're putting out. Yeah, because that's the question.
Thank you.
Okay. Thank you very much. Thank you, Pinaki, for handling those online questions. And thank you so much for our speakers and panelists of session 4. At this time, I'd like to welcome Markham to the stage again. Thank you so much.
All right. So let's hear once again our OPQ colleagues and our colleagues from CDRH and industry joined us in this wonderful talk about quality and reliability. So thank you, guys. There was quality built into that panel. Hello. So my job here is to close out the virtual part of this hybrid meeting, and then what we're going to do is then delve into the in-person part of this meeting. So I want you to all help me with this first. So what we're going to first do is to thank all the folks who had something to do with the behind the scenes work that we did. So this is all part of CRCG. Jim and Anna, along with Sam, you all are working together. This is a cooperative, and this is Jim's show, I understand. Anna did part one. So Anna was at forefront, the first hybrid meeting, and this is Jim's doing the second hybrid meeting for CRCG. This is excellent work, setting the leadership for this. This is all Catherine's group, Team D in my division. Catherine, you and Betsy and Karthika and Katie and all of you did a wonderful job working together to set the vision for what is 101. What do we want to hear? What do people need to succeed in this environment for combination products and generic drugs? I wanted to thank Maria for her work in project managing the session. Wanted to thank Jen and Jeffrey and Visha from University of Maryland. You all right there back in the booth, you see them? And Visha's out there with the conference room. Y'all did wonderful, a seamless AV work there, fantastic. And then I want to thank our industry planning committee members, Brandon Wood and Rasheen. Rasheen isn't here today, but I hear she'd had wonderful contributions to this session. So thanks to both Brandon and Rasheen. And so once again, thanks to all the planning committee. I didn't mention your name. I want to include you in that. So everybody on the planning committee, thank you. And then we're going to delve into a little recap of what happened today. So Jim opened up with CRCG. It's our public-private partnership, working with FDA industry, getting communications across the gap or chasm between us, and working together hand in hand collaboratively to foster a good generic product development. Wonderful example of what CRCG can accomplish in this space. So really appreciate that. And Jim opened up with some highlights from what we did before COVID, during COVID, and now that we're coming out of COVID, what we're doing now. So thank you, Jim. And then Rob opened up with a wonderful talk about the vision behind what we're doing here with combination products. This is a new space. This is where bringing combination products is come to the forefront. I remember when the first time I heard combination products was at the turn of the century, like around 2000, Mark Kaplan came on board and we were saying, okay, well, we have to do something about these devices that work with drugs. And here in generics, we're focusing on drug delivery products. These are devices that are the sole product purpose of many of these devices is to deliver drugs. There are some devices that are utilized that don't deliver drugs, but they modify the drugs. So these are the lights and things like that. And we didn't really talk a lot about that in the context of here, but some of the general principles do apply there as well. But Rod gave a wonderful overview of what we're trying to do in this space from the Office of Research Standards perspective, from the OGD perspective, and opened up a wonderful opening to this session. Part one, we Kim gave an introduction and highlighted some of the challenges that we have with identifying what are drug device combination products. It isn't always that obvious, but some of them are very obvious, but some of them are less so obvious. And so how do we do that? and you heard in detail discussion of some of the nuances about how to identify drug device combination products. Session 2 was headed by our wonderful Catherine Hartka. Catherine, wonderful job. Catherine is relatively new to our office, and she came from the Center for Tobaccos. She's a pharmacologist, a PhD PharmD. Great job, Catherine, on working together with the panel, industry, and everybody to present about drug device combinations and what are comparative analyses we do. And we talked a bit about there during that session about what are the best practices in getting together information to put into your ANDA submission and preparing for that. What resources available to you in the pre-ANDA space to help guide industry to getting a good submission for an ANDA with regards to comparative analyses. And also what to do when you comparative analysis show other minor differences. Do you then need to do a study? Or are there other ways to approach that? And then the last session on the comparative piece was from Catherine Fibus. And Catherine, you led that session where we talked about what are the studies that we need to identify these other design differences. How do you examine the impact of these products? We learned a lot from that session. We had our statistical colleagues. We had our colleagues from the Office of Clinical OSCE, Office of Surveillance and Clinical Effectiveness. So those folks have come and helped us with that. So thank you, guys. And then we had our lunch. Thank you for a wonderful lunch for everybody. Hope you all appreciated that. We had a chance with our first session to test out the different lunches. We decided this was the best lunch. So you all benefited from that first trial. And then we had our session with our quality colleagues. And that was wonderful discussion about what we're looking at, what FDA looks at when product is submitted. So that's I think a good highlight. If the folks want the tapes and recordings will be available and they'll be posted on the CRCG website. Should you want to delve back and pull up specifics, and we're not going to put together a blooper reel, but you can go and look and see how those sessions went yourselves. And then what we're going to do is for this afternoon, we're going to break out into some specific in-person sessions. We're going to be doing a combination product development simulation that's going to be headed by Betsy. So we're going to be hearing more about that for the folks here in the room. You're lucky to be here in this hybrid meeting, physically present, and we're going to get some energy going here. So there is caffeine over there. Should you want to enhance your energy a little bit with sugar and caffeine there, and any questions, etc. Catherine, thank you. Yeah, thank you. Let's hear it again for all the preparers. Thank you. Thank you. Thank you.